In this video lecture, we're going to go over Chapter 6, Part 1, the muscular system. And when we think about muscles, what is the main function we think of? Good, movement. And so in our body, we have three different types of muscle tissue. We have skeletal muscle, cardiac muscle, and smooth muscle tissue. Now, if we look at smooth muscle tissue, this is actually unique because smooth muscle tissue, we find this lining our organs of our internal digestive tract. Also, anytime we can move things where we don't have any control over them, this is really going to be our smooth muscle. And our smooth muscle is arranged in two different layers. We have an inner circular layer, which we see over here, and an outer longitudinal layer, which we see over here. And it kind of looks funny. How is this longitudinal? Let's draw a tube. And imagine this as this long tube of our digestive system called the alimentary canal. And we have this inner layer, circular layer, because it kind of looks like a circle. As we can see here, all of these cells are forming and going along this inner circular region. But the outside, here is each individual smooth muscle cell. If you imagine each of these lining up like this is why it's called an outer longitudinal layer, is because it's longitudinally placed. Okay, and so that's what it looks like with all the nuclei in the inside. And so when you do a cross section of this, cutting it in half, this way, what happens is that you cut the cells directly in half. So here's one cell. We're cutting it in half this way. And when we look at it from the side view, this is what we see. So this is a cross-section view. Now it's a good thing that we're going over this right now, because when we go over Chapter 14, the digestive system, we're going to go over these two layers again. So it's a good thing to kind of get familiar with it now. Now, we went over those three different types of muscles very briefly, but now we're just going to take a moment to compare and contrast these different muscles. And so if we first look at skeletal muscle versus smooth muscle and cardiac muscle, what do we notice about the number of nuclei? Okay, so we notice skeletal muscle. Okay, let's actually erase this really quick. Okay, so let's go skeletal, smooth, and cardiac. Number of nuclei. Here we actually have multiple, that's good, and they're all lateral, right? So we have many nuclei. What about here? Here's an example of one smooth muscle cell. Good, we only have one nucleus per cell. In cardiac muscle cell, what about that? It's one, but sometimes you'll see it in two, so 1 to 2 nuclei per cell. Now what about function? So this one, oops. Okay, well, so you get the idea. So what about function? What do we know about skeletal muscle versus smooth versus cardiac? Okay, anytime you can voluntarily contract your muscles, so that's now I tell you to flex your biceps, right? So you can easily move your forearm towards your humerus. Okay, so creating that movement, anytime you can voluntarily tell your muscles to contract, that's going to be skeletal muscles. So movement. So skeletal is always going to be under voluntary control. What about smooth muscles? Smooth muscles, these are the muscles lining your respiratory system, your digestive tract, all of your blood vessels within your arteries, your veins, and your capillaries. Do you have control over that? No, you don't. So these are all involuntary. And cardiac muscle, where do you find this? In the heart. That's why it's called cardiac. So these are all the muscles lining the walls of the heart. So is it voluntary or involuntary? Involuntary. Great, and we have something called a pacemaker within our heart, and that's going to set off a series of when your heart muscle is going to contract. And so because of that, it's all under involuntary control. Now other things we can look at are, what about the shape? So skeletal muscles is this nice cylindrical, long shape. So long and cylindrical. And then smooth muscle, what is that? So this is actually called fusiform or spindle-like. And cardiac muscle, we give a special term called branching. Now, other things you should note are the presence of these striations. And so skeletal muscles, we have these nice striations, kind of looks like those stripes. So long and cylindrical. Okay, we're going to clear this out and we're going to do another column again. So we had skeletal, cardiac, and smooth. And so characteristics. Skeletal, we said it has striations. What about cardiac? Also has these nice striations. And then finally smooth, we're giving that term smooth because it has no striations. Other characteristics we see? Here you can notice intercalated discs found in cardiac muscles, and these allow the cells to communicate with each other. So intercalated discs are what type of junction? Good, gap junctions. So these are gap junctions. In the presence of intercalated discs, which serve as gap junctions, and because of these gap junctions, we can have a coordination of contraction from one region of the heart and another region of the heart. And this is really key. So this will always be found only in cardiac muscle cells. Okay, so let's go over the comparison one more time of all the different types of muscles and what's characteristic of them. So body location, we mentioned. Body location for skeletal muscle, anytime you can voluntarily contract a muscle. So these are going to be attached to bones, some facial muscles, and also your skin. Cardiac, because it deals with the heart, it's always going to be the muscles lining the heart. Smooth, these are visceral organs, or organs we have no control over, part of our digestive system. Looking at the cell shape and appearance, we have cylindrical, multinucleated, here are the nuclei, cells with very obvious striations, branching, uninucleated or uninucleated, so one or two sometimes nuclei. We do see striations, and of course characteristic of your intercalated discs. And then finally single, these are smooth muscles are single, so one nucleus per cell, fusiform, and no striations. Now, regulation of contraction, skeletal will be voluntary, and because it's voluntary, we can either have slow or fast contractions depending on whatever you voluntarily decide you want your muscles to move or how you want them to move. And because of that, there's no constant rhythmic contraction. It's all voluntary, so you can tell it to speed up, slow down, and when to contract. Cardiac muscles, these are all involuntary, set off by a pacemaker in our right atria. This is slow contraction and it's rhythmic. It's always set off by that pacemaker. Smooth muscle, involuntary, and it's all based on our nervous system control. So whether we're going to be ready for fight and flight or rest and digest systems, that's going to determine how fast our respiratory system organs and then also our digestive system organs are going to be moving. This is under slow contraction, very typical. And is it rhythmic? In some, but not all the time. So different areas of this where smooth muscles line are not going to always be consistent when they're contraction. Now let's go over category by category these different types of muscles, starting with the skeletal muscles, and how are muscles attached to bones because we have all that voluntary contractions. As a review, good, they are attached by tendons. And the function of skeletal muscles in general is, the first thing is to maintain your posture. As you contract your latissimus dorsi, your dorsal muscles, you can bring up your vertebra and also your thoracic cavity. So in that sense, maintaining your posture. You can also stabilize your joints. And the most important thing, which is the only muscle to do this, is to generate heat. And remember, shivering, as you shiver, you're constantly moving your skeletal muscles, and as they contract, they break those ATP bonds and then release heat. Now, last year, I took a picture of my Kailua cabbage pork plate, and I was pretty fascinated. We're talking about the skeletal system, and it's this, sometimes you don't realize, but you stop and think to kind of think about the food you're actually eating. And of course, in a Kailua pork, you notice here are all the muscles of the pig, right? I dissect my food as I eat it. And you can notice that you can notice sometimes when you eat pulled pork, some of the pork falls into small pieces, but also some of them remain as these larger chunks. And why is that? It's because of the anatomy and the shape and how these muscle cells for the skeletal muscles are all grouped together. Okay, so let's go over the anatomy of all these muscles. And so the first thing we notice is that the very smallest unit of the muscle cell is called a muscle fiber. And so we can use that synonymously. So one muscle cell is also known as a muscle fiber. And before, when we looked at cells in chapter 3, we mentioned that typically you imagine them as spherical or oblong, but cells in fact have different sizes and shapes. And here with a muscle fiber cell, it's that nice long cylindrical shape with lots of nuclei. And so that's exactly what this is over here. Now we take one muscle fiber, or one of these cells, you couple them with many other muscle fiber cells, and you can kind of wrap them around. I think of it as tape, but really this is more of like a connective tissue. And this connective tissue is called a paramecium. Now a paramecium groups all these muscle fiber cells together, and once you bunch them up together, what you form is called a fascicle. So that's the first layer. Now the next thing you can do is take a bunch of these fascicles, you group those together, lots of these fascicles together, you wrap it around another layer of connective tissue. And this connective tissue that you wrap around is called an epimecium. So the epimecium will bunch all of these fascicles together. Now one more thing we have is actually an endomecium, which I forgot to mention. And these endomeciums are between each of these muscle fiber cells. So here's one of your muscle fiber cells, and we're going to wrap that up in that endomecium, and then endomecium and those muscle fiber cells, we wrap all those together and we wrap that around with a fascicle. Now, with this epimecium is going to continue along with all of the paramecium and endomecium. All that's going to continue to form this tendon. So it's an extension of all of those coverings, and the tendon is going to attach to our bone. And so really this is the anatomy of how we can attach all those muscles, those single muscle fibers, to our bone. Now, epimecium, we just mentioned, will blend into that connective tissue attachment, forming the tendon, which is that cord-like structure that we see attaching the muscle to our bone. It's mostly composed of collagen fibers, and it will often cross a joint to toughness and its small size. Now, another thing we have attaching muscles to bones or muscles to other muscles, it's called an aponeurosis. Aponeurosis. And this is more of a sheath-like structure, so this looks like a cord where it bunches up. This one's more of like a sheet, so it's very flat, a broad tendon. It'll attach your muscles indirectly to bones or cartilages using these connective tissue coverings. And a great example is right over here. The anterior abdominal aponeuroses are located just superficial to our rectus abdominis. And here is our rectus abdominis. Here is that abdominal aponeurosis. And see, as you can notice, it's not directly attached to our bones, but it helps connect muscles to each other. And so that's why it's indirect attachment to bones, cartilages, or other connective tissues. So we have external oblique, which is this one over here as its border, pectoralis major as its superior border, and latissimus dorsi, which is on the opposite area. So this is more dorsal. So that's how we can connect those together. Now, other things we have looking at our microscopic anatomy of skeletal muscles, if we take a closer look and you take a cross section of one of those skeletal muscles, muscle fibers, and you take it a look closer, you'll notice that in each of our cells, and each of these muscles, so this is a muscle fiber, within our muscle fiber, we actually compose of smaller structures called myofibrils. Okay, and each of these myofibrils, these are long organelles inside each of these muscle cells. And so within our normal cell, that we can consider as normal cell, right, we have our nucleus, that's over here, Golgi apparatus, rough ER, smooth ER. Here, majority of these organelles are called myofibrils. And then surrounding each of these myofibrils, this is the main organelle, we have something called the sarcoplasmic reticulum, and that's here in blue. Now, the function of the sarcoplasmic reticulum is that it stores calcium, and this is very important because calcium we need for our muscles to contract. Okay, here in our typical cell, we have this plasma or cell membrane. Here, within our cell, we also have a plasma and cell membrane, but we have a special term for it, and we call it a sarcolemma. So sarcolemma is a specialized term. Okay, and so of course I took more pictures of that meat that I had, and here is one of those myofibrils found within our muscle fiber cell. Here are lots of those fascicles. So here's one fascicle, here's another fascicle, so there's another fascicle, here's another fascicle, okay? And you can bunch those all together. Now before we go over how muscles contract, we should go over a little more anatomy of our skeletal muscle cell. And I want you to notice over here we have these stripes and what actually composes those stripes. So we have this dark region called the A band, and we have this lighter region forming the light part of the stripes called the I band. And what gives rise to these banding patterns, this dark A band and light A band, are structures called myofilaments. And these myofilaments span the entire length of our myofibrils. And so we have, here's one myofilament and here's another myofilament. And so really it's the movement of these myofilaments crossing over each other that give rise to muscle contraction. And so how do muscles contract? It's the shortening of muscles due to the movement of myofilaments in the myofibrils. Okay, so let's give more terminology to this. And so here we can start noticing the closer anatomy of all of those myofibrils and all those myofilaments in those myofibrils. And so here we go. Myofibrils are lined to give distinct bands. First, we're going to go over the I band. And I remember I because it's the light band, so I for light. And this contains only thin filaments. And here's another I. Okay, so I band, light band, which is thin filaments. And this is composed of the protein actin. And so here is actin. Everything that you see in blue is all of those thin filaments. And how do we hold these in place? They're anchored by the structure here called the Z disc. Okay. Now more anatomy. So this is that light white banding structure that we see. Now going into this dark banding structure that we see here in red, this is called the A band. This gives rise to this dark region. So here's that stripes, here's that light stripe. Okay, and this contains the entire length of the thick filaments. So you notice how they're much thicker, they're much wider compared to those blue bands. It's composed of the protein myosin, and it's anchored to the M line. Now what's also important is that these dark bands, they compose, all these filaments have heads that have extensions that are able to kind of walk along these thin filaments, and we'll see that what that looks like in a little animation. So we just need to make a note that the myosin heads will walk along the actin, and that in turn will give rise to shortening of the myofibril. Now, a lot of terminology, so it's a good thing to go over these over and over again. Okay, and so it's kind of hard to show you without me physically showing my hands to you, but let's just imagine. I want you to put your hands out. 1, 2, 3, 4, 5. Okay, and so here are your fingers, and you can imagine your fingers as myosin. And here you can also imagine if there's just this line, this is going to be that actin. Now what I want you to do is spread your fingers apart and slowly start bringing them together until they cross over. 1, 2, 3, 4, 5. Yeah, I know your fingers don't really look like that, but let's just imagine that they do. 4, 5. Ooh, I guess you have a fused thumb and index finger. Okay, and what you notice is that as you overlap your fingers together, what do you have now? You're bringing in and you're shortening this area over here. And really, this is how the muscles will work. When you can relax, when your fingers are farther apart from each other, this is when your muscles relax. And once your fingers come closer together, that's myosin moving along actin. And this overlap that you create is when those muscles begin to contract. So contraction deals with this overlap that you create with myosin and actin. Now, let's look at a closer look of what this myosin head looks like. Here's actin, all that thin filaments. Here's myosin in red, and here's all of those heads on that myosin. And you can imagine them just walking along this actin filament, and as they walk along this actin filament, actin and myosin cross over more, and that gives rise to shortening of your muscle. Now the way that all these myofibrils are organized, they're organized into sections called sarcomeres. So here's our Z disc anchoring down our actin, M line anchoring down our myosin. And so it's really the sarcomere extends from one Z disc to another Z disc, and this overlap that you create, a shortening, will also correlate to that contraction. So a sarcomere is a contractile unit of the myofibril. And let's look at more illustrations of what that means, and hopefully we get a better illustration of that. Okay, so here we have our Z disc. Here we have our other Z disc. So here's one and here's one, and we have our M line. So here is one sarcomere, and here's our second sarcomere. Another Z disc, two Z discs with our M line in the middle. Now this is a relaxed muscle. And in a relaxed muscle, we have no overlap of our actin and our myosin filaments. As you can see here, they're pretty far apart. Now what happens in a partially contracted muscle, myosin heads are going to start walking over our actin filaments. And what happens to that? We have shortening. We have more of this overlap that we see with actin and myosin. Let's look at an image over here. So here's relaxed. Not a lot of overlap. An overlap we see as these darkened area over here. Here we have partial contraction, as we notice Now we have a wider dark region, and that's because we have more overlap over here. In the fully contracted muscle, our myosin heads are going to walk clearly all over this actin filament over here. So we have complete overlap, and this is that shortening of our muscles. Now this is a fully contracted muscle. And as we can see here, we have that complete overlap, hence why we see a much wider, darker region. Okay, and so when we can notice, and we can also regulate how much of this overlap occurs by looking at different zones and different bands. So the first area I want you to look at is called the I band, and the I band is a zone of thin filaments that are not superimposed by thick filaments. Filaments not superimposed by thick filaments, meaning it's the area where there's no overlap. And so so it's all the thin filaments where there's no overlap of those dark or thick bands. What do you expect the width of the I band to happen over time from a relaxed muscle to fully contracted muscle? Good, so over time, your I band should shorten from a relaxed muscle to a contracted muscle. And we see that here. So with more overlap, the area where there's only thin filaments is going to start decreasing. Now the next area I want you to look at is the A band, which is this region over here. And the A band is the length of a single thick filament. Now this is kind of tricky because it's only the length of the thick filament. Regardless of whether there's overlap or no overlap, it'll still be the same length. And so A band, whether it's from relaxed to a fully contracted muscle, it will not change. And so no change over time. Okay, and so that's very key, really important too. Okay, so this is the only one that will not change over time. Now the last one is called the H zone, and the H zone corresponds to the zone of the thick filament not superimposed by the thin filament. Okay, so that's over here. So here we have only the thick filament. Let's write that down first. So zone of thick filament not superimposed by the thin filament. Okay, so here we can see relaxed muscle, we have no overlap. So superimposed means no overlap. Here we have no overlap for thin and thick. Partially contracted, now we have more of that overlap with thin and thick, and because of that, our region of overlap is starting to get decreased. And finally, in a fully contracted muscle, we have complete overlap of thin and thick filaments, therefore this decreases as well. So keynotes, both the I band and the H zone, those are going to decrease, and H zone, shorten from a relaxed to a contracted, and the A band is the only one that's going to stay the same. Now there's another region that I want you to see, and that's within this A band. So A band is the entire length of your myosin filaments. There is a region within this A band where there's not going to be any actin filaments at all, and that's going to be called the bare zone, and we also call that the H zone. Okay, so that's what we saw over here. So just two different terms for that H zone. So it's H zone or our bare zone. Now for the next couple of slides, we're going to go over how skeletal muscles contract, and it really deals with all these main structures, the terminal cisternae or the transverse tubule, and the sarcoplasmic reticulum, as well as all of our myofibrils. And so really what's going to happen, as we are going to see, is that a signal is going to come down to cause our muscles to contract. So we're going to send that signal down the length of our entire sarcolemma, and that's going to cause the release of calcium from the stores in our sarcoplasmic reticulum to get released into all of our sarcoplasm. So sarcoplasm, this is the cytoplasm of our muscle fiber cell. And so that's going to get released into our sarcoplasm, and that's going to initiate this crossover of both our actin and myosin myofibrils. And before we get into that, let's go over a few properties of muscle fiber cells. And the first one is that they have excitability, or responsiveness or irritability, meaning that they're able to receive and respond to a stimulus. And that stimulus will stem from our central nervous system, and that's going to cause our muscles to contract. Now, we also have the idea of contractility, and so once they receive that signal, muscles will be able to shorten when that stimulus is received. Stimulus is received. And then finally, or sorry, not finally, but another property they have is extensibility, meaning that muscle cells can also be stretched. And then finally, elasticity. This is that once we've stretched or contracted our muscles, they are able to recoil and resume their resting length. Now let's look at the stimulus. Where is the stimulus coming from that's going to tell our muscles to contract? And so really our skeletal muscles must be stimulated by a nerve impulse in order for that to contract. And all these nerve impulses are going to stem right here from our brain and our spinal cord. Okay, so stimulus from a nerve impulse. From central nervous system. Okay. So let's go over a couple of things. And so first here, once we receive that signal that we want our muscles to contract, we're going to send that message down here through our spinal cord, and we have something called a neuron. And now neurons, we're going to learn about these in chapter 7, so we're kind of going a little ahead. But neurons, basically what they do is that once they receive a signal to contract a muscle, they're going to send that stimulus down this region. This is called an axon. And so this axon can send a signal all the way down from our spinal cord to our muscle fiber cells. Now, once the stimulus is strong enough, that in turn is going to rush down the entire length of our sarcolemma and then cause the release of all those calcium stored, and then the muscles will contract. And so what we have here are two things that we see in this illustration. The first thing is a motor neuron. And these motor neurons, these are those nerve cells that are located in the spinal cord. And their axons are going to extend to the muscles. Now another thing we see here is called a motor unit, and this motor unit that you see here, this is comprised of our motor neuron and the muscle fibers that it innervates. And what we'll notice is that here we can have a stimulus from one neuron, and that can innervate this region of this muscle over here, and also this region of other muscles over here. We can also have other neurons that innervate the same muscle but a different region over here, and over here. And now what you can also have is stimulation, so you can have both of these motor neurons stimulate at the same time. And once they do that, that will allow this entire region of that fascicle to contract those myofibrils at the same time. And that's how you can have coordination of contraction of a certain region. Now, I know some of you might be thinking, what about a twitch? Well, a twitch can occur if, let's say, if one of these motor neurons go off, it can cause contraction of some of these muscles and not the entire region. So really, twitches are irregular contractions. They're just kind of a stimulation from your central nervous system to tell your muscles to contract. Okay, now I want you to think about, we just had this axon, and here we have the ends of those axons, and they're actually not going to touch our muscle fibers. They're not going to touch our sarcolemmas of our muscle fibers. We actually create this small little gap between them, and because we can't touch them directly, we have to have different chemicals that can move from the end of our axons to the beginning of our sarcolemma. And so this entire region that we're going to look at, this is called the neuromuscular junction. Neuro dealing with our central nervous system, muscular because it deals with our muscles. So neuromuscular junction. And this is the axon terminal. So remember, this is our axon. Terminal meaning the very end of it. So axon terminal. And the sarcolemma of the muscle cell. And so here's that sarcolemma. So axon terminal and sarcolemma of the muscle cell. And because we can't communicate, what do we need? Now we have chemical messengers, which we call neurotransmitters, and these are found within vesicles. And so we have neurotransmitters that will move. That will be released by or from vesicles in the axon terminal and will bind to receptors on the sarcolemma. Okay, now what are these neurotransmitters called? Because we're dealing with skeletal muscles and we're dealing with the contraction and movement of these skeletal muscles, the specific neurotransmitter that we're talking about is acetylcholine. And we can abbreviate that by ACH. Okay, and so I want you to pause this video right now, and here's this video link for the events at the neuromuscular junction, and it's going to help explain what's going on in this region. And we're going to go over it step by step, but it's always a good idea to watch the video and then we're going to talk about it. And I also have this link for you on your LaLima page. Okay, now after you've watched that video, let's go over exactly what we saw. And so what we saw was here we have our spinal cord. It's going to send that message to contract down our axon. Here's our cell body of our neuron. Here's our axon. And this right here, this is once again the what? Axon terminal. So that's labeled over here, axon terminal. Quick review, we have these vesicles found in our axon terminal. These are filled with what neurotransmitter? Good, acetylcholine. Other things that we have here, well, we have, because we don't touch directly our axon terminal with our sarcolemma, we have the space, and the space is called the synaptic cleft. So the synaptic cleft is a space between the axon terminal and the sarcolemma. Here, this is a membrane, the sarcolemma. Okay, and then finally also we have here is this calcium that's going to enter into these protein carriers, through these protein carriers, and then enter here into our axon terminal. Now down here on our sarcolemma we have a receptor, and this is a receptor for what? Good, it's our acetylcholine receptor. And right here in green, what is that? That's going to be our acetylcholine. Okay, now that we've gone through everything, let's go step by step of what happens. And so the very first thing that's going to happen is that we're going to receive that stimulus from our central nervous system, and that's going to initiate the very first steps of how we can finally eventually have this muscle contraction. So it's going to reach our axon terminal of our motor neuron. Once it reaches our axon terminal, what's going to happen? It's going to initiate calcium to enter into our axon terminal. And what does calcium do? Calcium will cause the synaptic vesicles to fuse here with our axon terminal membrane. And what does it do when it fuses? It'll release all of this acetylcholine into our synaptic cleft. So you can see it's starting to get released over here. Where is that acetylcholine going to go? It's going to bind then to our acetylcholine receptors. Now once this binds to our acetylcholine receptors, now we can initiate, depending on if there's enough acetylcholine and enough stimulus of that acetylcholine, it'll initiate the steps to actually have a muscle contract. Okay. So now, in response to the binding of acetylcholine to the receptor, what did you notice from that video? The sarcolemma becomes permeable to what ion? Sodium. Good. And how do we get this more permeability to sodium? Okay, so let's go over this really quick. And this is that sodium-potassium pump that we saw once before. And we're going to go over exactly why this is important, especially for this impulse to occur. In our cell at rest, in our muscle fiber cells, we're going to have a high amount of sodium outside of our cell. Inside of our cell, we're going to have a high amount of potassium. And how do we maintain that? Through our sodium-potassium pump system. And how does this work? Well, at rest, we're going to have some sodium because we have lots of concentration of sodium outside of the cell, lots of potassium inside of the cell. By simple diffusion, where does sodium want to go? Sodium wants to go inside. Good. Simple diffusion, where does potassium want to go? Potassium wants to go outside. Great. Now, if we want to maintain high sodium outside and high potassium inside, what do we need to do? We need to use a sodium-potassium pump, and we got to use energy. Energy because we're going against our concentration gradient. And so the sodium-potassium pump, what's it going to do? It's going to take the sodium that's came in, it's going to use energy, it's going to break that ATP into ADP and that phosphate. By breaking that, it's going to release energy. Energy is going to drive this to move all that sodium out. In exchange, it's going to bring potassium into our cell. And once it brings potassium inside of the cell, then we can maintain this gradient. And because here's our sarcolemma, outside we have all of that sodium, inside we have an overall negative charge. And this negative charge is because of this potassium inside, sodium outside. We also have large proteins, and all these proteins are going to carry a negative charge. So the overall charge of our membrane is going to be very negative. Now what's going to happen is that because sodium, our membrane, once we receive that signal, our membrane is going to be more permeable to sodium. Sodium is this highly positive charge, so what's going to happen over time? So once our acetylcholine binds to all of these receptors found in our sarcolemma, sodium is going to come rushing in, And what's going to happen to our membrane? Our membrane is going to become more positive. Okay? And this positive charge is going to start sweeping across our entire sarcolemma. So let's see what happens to that. Okay, so here we have acetylcholine is going to bind. It's going to allow these channels, ion channels, to open up because we have high concentration of sodium outside of our cell, high concentration of potassium inside of our cell. Just by diffusion, sodium is going to come rushing in, potassium is going to go rushing out. And that's going to cause our what's called a depolarization, a localized depolarization. We're depolarizing because we're changing this membrane that was once negative to now becoming positive. And so we're going to initiate that right over here. Now what's going to happen? I'm just going to skip that really fast. The moment that this membrane becomes depolarized, it's going to cause this region to be, if there's enough signal and it becomes more positive, it's going to cause this entire membrane, kind of like a flame igniting a twig, spreading across the entire branch. Same thing's going to happen within our muscle fiber cell. This region here is going to become positive, but it's going to easily start sweeping positive in this direction over here, and that's called an action potential. It's going to spread across, make this entire region from negative to become positive. And once it becomes positive over here, eventually the side will become negative once again. And why is this important? Because this is going to initiate the release of calcium from the sarcoplasmic reticulum. And that in turn, once calcium is released, then we can have this contraction and interaction of the myofibrils. Giving rise to contraction. So let's get a step back and let's see how we can stop this. And so we'll get into a little bit how contraction occurs, but there are two ways that we can stop this depolarization from occurring. The first thing is if this acetylcholine gets broken down, so it can get degraded by acetylcholinesterase. Anytime you see an ACE, this deals with an enzyme. So acetylcholinesterase is going to degrade acetylcholine. So that's the first way. So how to stop an action potential. First thing, you have acetylcholinesterase. That's going to degrade acetylcholine. And the second thing that can happen is just simple diffusion of acetylcholine out of the synaptic cleft. So two different ways. Now let's go over how this action potential or depolarization of our sarcolemma will eventually give rise to our muscle contraction. And before we do that, I want you to first pause right now, pause the video, and watch this YouTube link and this YouTube video right over here, also found in LaLima, of how contractions occur. So what I want you to see here, we have stemming from, we'll do a quick review, our spinal cord is going to send a message to tell our muscles to contract. We're going to receive that signal at our cell body of our neuron. We're going to send it down our axon, and it's going to be received here at our axon terminal. Once it's received at our axon terminal, we're going to have ions coming into our axon terminal. What ion is it? Good, calcium. Calcium is going to cause what to happen? It's going to cause these vesicles to fuse with our axon terminal and release these neurotransmitters into our synaptic cleft. Which neurotransmitter are we looking at? Good, acetylcholine. So acetylcholine is going to bind to acetylcholine receptors found along our sarcolemma of our myofibril. Okay, good. So make sure you're clear with that so far before you move on. Now, what's going to happen? Once acetylcholine binds to our acetylcholine receptors, here we have a high sodium gradient outside of our cell, Inside of our cell, we have high potassium. Where's sodium going to do? It's going to rush inside of our cell. Oops, I should make this span our entire membrane. Sodium's going to come into our cell, and then potassium's going to go outside of our cell. Initially, our membrane is going to be negative, but because sodium comes in, we change it to a localized positive charge. Now, if that stimulus is strong enough, if we can make this very positive, what's going to happen? We're going to lead to an action potential. An action potential, this is a message, it's a positive charge that's going to sweep across our entire sarcolemma. So it's comparable to that twig getting set on fire. Spread our entire sarcolemma. Now, along our sarcolemma, we have these invaginations of our cell membrane, and these are called T-tubules or transverse tubules. And what is the function of this? This is found, what's adjacent to that inside are all of our sarcoplasmic reticulum. And what's important? Well, this is important because it carries and stores what ion? It stores calcium. Now, you notice you have calcium entering into your axon terminal. That's part of our nervous system, so our motor neuron. Inside our muscle fiber cell, you also have another calcium. Make sure you know the function and purpose of these two calciums and what they lead to downstream. So, we have this calcium. It's get released by our sarcoplasmic reticulum. This entire, everything here in pink, this is our sarcoplasm. So it's just like the cytoplasm of our myofibril. And this calcium, what's going to do? It's going to flood our entire inner sarcoplasm. And now it's going to bind to blocking proteins on actin to expose the binding site for myosin to bind. So calcium binds to blocking proteins on actin, which will then expose the binding site of myosin, for myosin to bind to actin. Okay, so here we have actin. We're going to bind calcium, as you can see over here. We're now exposing the binding site, so everything here in yellow now we can expose to these myosin heads in red. We have that nice connection, and then eventually we have with ATP, only in the presence of ATP, ATP can then charge up your myosin heads and then walk across actin. Now, how do we stop contraction from occurring? What are those two methods again? Good. We're going to have to stop it over here. We got to stop it from this source. If we remove, so you can remove acetylcholine either by diffusion out of the cleft or remove acetylcholine by that acetylcholine esterase. Now, how does that work? Well, you stop acetylcholine from attaching to your receptors, and what happens? Sodium will not go through. Our membrane becomes negative once again. You don't have your action potential. Without your action potential, you're not going to release calcium from your sarcoplasmic reticulum. No release from your sarcoplasmic reticulum. You can't continue the exposed binding site. So what happens? This is going to stop. Calcium is going to get sucked back into our sarcoplasmic reticulum. We are now covering all of these binding sites. So now actin is closing off and shutting down and saying, nope, no more binding for myosin. Myosin has nowhere to bind. Myosin relaxes, and that in turn causes the muscles to relax. Now I want you to listen to this video again, also read it in your textbook. Go over this because this is very important. It's very especially important because we're going to cover this on your next exam. Now, other things. So we mentioned troponin, tropomyosin, a lot of these other terms, troponin, tropomyosin complex, this stuff. You don't need to know this for your exam. All I want you to know is actin and myosin and how we can expose the binding site and then remove the binding site. And these two mechanisms to stop contraction from continuing. Let's go over one more time how this contraction occurs. And here we have our actin, here we have our myosin, and once again this complete overlap is at full contraction of the muscle. Here's a closer look at that actin and myosin. Blue is the actin, and red is the myosin, since we didn't get a good look 2 slides ago. Here we have blocking proteins where myosin wants to bind to actin, and it's all both protein complexes, which we don't really need to know about right now. Here is our sarcolemma, and here is our transverse tubules in our sarcolemma. And adjacent to that, remember, we have all of our sarcoplasmic reticulum. Housing which ion? Good, it houses calcium. So under normal conditions, our membrane is negatively charged. However, with our action potential, once all its sodium ions come into our cell, our membrane becomes more positive, which in turn causes a release of calcium ions into our sarcoplasm. So here is those calcium ions being released. What does calcium do? Calcium binds to all those blocking proteins, and then now we'll expose the binding site for myosin to bind to actin. Okay, so what happens then? Well, myosin is now, once those binding sites are exposed, myosin can bind to actin. However, to continue this walking action across the actin, you need a new ATP molecule. So the only way to disengage actin from myosin is for ATP to bind to your myosin, a fresh new one. So once this binds, then we have a release of myosin from actin, and then we can break ATP into ADP and phosphate once more to activate the cycle again. And eventually, with more of that ATP and continued exposure to those calcium ions, then we can have further contraction. Now here is a closer look at that cross-bridge cycle. Here's a link to watch that animation, so I'll pause right now for you to watch that, and we'll go over that once again. Okay, now that you've watched it, let's go over the couple of steps. Here we have tropomyosin and troponin. These are both blocking proteins on actin to prevent myosin from binding. And this is in a resting sarcomere. So here we see all of these are blocking, and here we have myosin head can't bind to our actin binding site. Now here, once calcium comes in, calcium's going to bind to the troponin, tropomyosin complex and expose now, here's all that actin for a myosin head to bind to. We have the first stage, or the first step, is a cross-bridge formation. And cross-bridge is really this interaction between myosin and your actin. And then we have movement. So we have myosin reactivation. Once this cross-bridge forms, this myosin head is going to walk across, pushing along this actin filament. And only in the presence of ATP can we detach myosin from our actin. Now that we've detached myosin from actin, ATP is going to break apart again to form ADP and phosphate. This is going to restart the entire cycle once more. And once again, this is only possible in the presence of calcium, and also continual contraction will occur as long as you have ATP as a source. Okay, so here is a summary of the entire contraction process. Let's start from the beginning, and it's really important to kind of go over this and say it to yourself over and over and over again. That way it makes more sense. So we have a stimulus coming from our spinal cord, our brain and our spinal cord. It's going to tell us to contract our muscles. So we have this nerve impulse that spans from our cell body, and it's going to go along our axon. It's going to reach into our axon terminal. At our axon terminal, we have an influx of what ion? Calcium. Calcium is going to cause what to occur? It's going to cause the fusion of vesicles filled with what neurotransmitter? Acetylcholine, to fuse with our axon terminal, release the acetylcholine into, what is this region called? Synaptic cleft. Synaptic cleft. And the acetylcholine is going to bind to acetylcholine receptors. In turn, what ion is going to come in? Good, sodium is going to come in. and potassium is going to go out. All in all, this is going to create an action potential that's going to turn our entire membrane more positive. This is going to enter into these imaginations of our sarcolemma called the T-tubules or transverse tubules, which in turn is going to cause calcium inside our muscle fiber cells to release calcium. Calcium then is going to do what? Good, it's going to bind to those blocking proteins on actin and allowing now our binding site for myosin to attach to actin will be exposed. We can form all those cross bridges that we saw, and then we have contraction. So now we have myosin walking along actin, and then our entire span of our sarcomere is going to start decreasing. So with contraction, sarcomere will decrease or shorten. Now, what will end this contraction? Two things. What was that again? Acetylcholine's got to be removed by acetylcholine esterase, or it will diffuse outside of the membrane. Because of that, we don't have an action potential anymore. This is going to stop the stimulation and release of calcium. Calcium is going to get retaken up into our sarcoplasmic reticulum. Now, troponin trypomyosin is going to block all the binding sites on actin, so now myosin heads cannot bind to our actin, and then finally we have relaxation. Okay, so everything that I just mentioned and went over are all listed over here, so make sure that you can go through these illustrations once again, go through each of these steps and see if it makes sense to you. Now, what's also important about contractions of skeletal muscles is that muscle fiber contractions is pretty much an all or none response, meaning that it can either have enough stimulus to allow the contraction to occur, or there's not enough stimulus, in which case there won't be any contraction. And also what's important is that within a skeletal muscle, not all fibers may be stimulated during the same interval. And we saw those with those motor neurons. So some motor neurons can innervate a region. So here's a motor neuron. Here's our muscle fiber cells. So some of them can innervate this region, another one can innervate this region, another one can innervate this region. Now, it depends on which motor neuron is stimulated in order to stimulate that region of the motor neuron, of the myofibril itself. So we're going to end off. I want you to go over these 4 review questions, or these 2 review questions and these 2 short answers, and we'll cover this at the beginning of next class.